Let us learn double beam UV-Vis spectrophotometers. Double beam UV-Vis spectrophotometers are instruments that measure the absorption, transmission, or reflection of ultraviolet and visible light by a sample, much like all UV-Vis spectrophotometers. However, they use two beams of light, one that passes through the sample and another one that serves as a reference. Spectrophotometers are instruments that measure the distribution of light through different wavelengths. Scientists utilize these devices to measure various types of light, such as visible and near ultraviolet light. In order to obtain measurements at a faster speed with greater precision, scientists have found that double beam spectrophotometer is a suitable solution. The principle of a double beam UV-Vis spectrophotometer. The fundamental principles underlying a standard double beam spectrometer are the dispersion, reflection, and the refraction of light. By employing these methodologies, it is possible to compute the results of analytes that exist in a biological specimen by relying on their absorption characteristics. Key features. That is nothing but the parts of the double beam spectrophotometer. Two light beams. The instrument splits the light beam from the light source into two beams, one for the reference and one for the sample. Monochromator. A monochromator selects a specific wavelength of light to be used for analysis. Detectors. Detectors measure the intensity of light in both the reference and sample beams. Electronic system. The instrument's electronic system processes the data from the detectors to provide output in terms of transmittance, absorption, or concentration. Microprocessor. Modern double beam UV-Vis spectrophotometers utilize microprocessor for data processing, enhancing accuracy and reproducibility. And this is the picture of a double beam UV visible spectrophotometer. So we have a source, and it can be either a tungsten or a deuterium, depending on UV or visible sample that we are going to have the characteristics. And this is a monochromator. It can be a prism or a grating, and here it is used as a prism. And we have a specific wavelength that comes from the spectrum of light because of the filter, and this is the filter. And this is split into two beams. One goes through the reference, usually a solvent, then another goes through the sample cuvette. And in the sample, light is absorbed, and again they recombine, and the detector detects the input, and we have the output. Either a microprocessor or a computer can display the output. So either it can be absorption, transmission, or reflection. Let us look into the working principles. A light source like a deuterium lamp or UV or a tungsten lamp are visible emits light. The light passes through a monochromator which selects a specific wavelength. The light beam is split into two beams. One is a reference beam, another one is a sample beam. Two beams are formed in the space by a V-shaped mirror called a beam splitter. One beam passes through the reference solution to a photodetector, and the second simultaneously traverses the sample to a second matched detector. The sample beam passes through the sample cuvette where it can be absorbed, transmitted, or reflected. The reference beam passes through a blank cuvette containing only the solvent. Detectors measure the intensity of light in both the beams. The instrument calculates the ratio of the light intensity in the sample beam to the reference beam, which is then used to determine absorption, transmittance, or concentration. The two outputs are amplified and the ratio is determined electronically or by the computer and displayed by the readout device. A double beam spectrophotometer is an analytical tool utilized for measuring the light absorption of liquid or gas samples containing graduated windows. Transmission in a double beam spectrophotometer is often accomplished by the utilization of ultraviolet radiation, infrared rays, and visible light. However, the more current and modern spectrophotometers make use of a broad spectrum of electromagnetic wavelengths in order to perform the reflection and transmission process. This spectrum of electromagnetic wavelengths includes visible rays, infrared rays, microwave rays, and ultraviolet rays. The determination of the amount of light that corresponds to a given wavelength is the primary function of a double beam spectrophotometer. In this stage of processes, the analyte present in the biological sample absorbs the beam of light. In spectrophotometry, the specimens, also known as samples, can be either gases or liquids. During the process of spectrophotometry, the analytes are mixed with the solvent and then allowed to dissolve. In order to prepare the solid pellets for usage as an analyte in the spectrophotometry, the solvents that are currently being utilized are first combined with a transparent matrix. When making the disc for the double beam spectrophotometer, a pellet press is used. This press has a disc that is suspended in the biological samples, and the light beams pass through the disc as they move through the press. The fundamental working principle of double beam spectrometer is predicted upon the phenomena of light ray reflection and transmittance. The primary component of both single and double beam spectrophotometers is the light source, which emits intense beams of light. The characteristics of these light beams are reliant upon the specific wavelength of interest, which may include electrically powered ultraviolet and visible rays as well as infrared rays. The monochromator effectively isolates light beams of a particular analytical wavelength from the broader spectrum of the light source. The selection of wavelengths is based on the spectral composition of the light rays that traverse the double beam spectrophotometer. The wavelength of the light ray employed in the instrument is determined by the absorbance properties of the analyte under consideration. Instruments known as monochromators have as their primary function and the facilitation of the passage of polychromatic light beams lies via the entry slit of the monochromator instrument. It also makes it possible for a single light ray wavelength that is monochromatic to depart the device via the exit slit. This fascinating and extremely focused beam of light are made up of a relatively small portion of the electromagnetic spectrum of light. The slit methods or apertures of the monochromator that is attached to the spectrophotometer are determined by the spread of the wavelengths as well as the electromagnetic band pass of those wavelengths. The light beams in the single and double beam spectrophotometer can be manipulated with the help of these monochromators, which include customizable settings. In this case, the process of transmission and reflection is aided by the characteristics of the light rays, which is what disperses the elements in the monochromator. The light source in the double beam spectrophotometer is utilized in the instrumental design to irradiate the samples with light beams. During the process of spectrometry, the light rays undergo a diversion of 90 degrees. The process involves the rotation of a disk containing three distinct panels within the spectrophotometer. One of the components of the apparatus facilitates the unobstructed transmission of the light beam through the disk without any incidence. And then another point consists of a mirror surface with the third point which is black. The direct illumination of a specimen cells occurs when the light beam traverses the disk panels. In the event that a sample under consideration is in a liquid or a gaseous state, it is housed within a cuvette that is composed of a transparent material which exhibits no absorption of light rays within the spectral range relevant to the double beam spectrophotometer. The substance of interest present in the specimen is solubilized in a solvent that is contained in the cuvette component of the dual beam spectrophotometer. When the incident light rays are reflected at a 90-degree angle by the rotating disk, they do not come into contact with a biological specimen. The incident light traverses a cuvette segment within the reference cell that exclusively comprises the solvent. This phenomena facilitates the propagation of electromagnetic radiation and the phenomenon of specular reflection. In a spectrophotometer, the light rays which do not get absorbed after traveling through every portion of the specimen cell or sample cell are directed onto the phototransducer or light detector. This occurs after the light rays have traveled through every part of the specimen cell or a sample cell. The arrival of photons in the light beams is translated into an electrical signal by this component's particular portion of the part. This conversion is carried out by using the screen of the computer. The spectrophotometer can be configured in such a way that it does not require the light pathways to be in a straight line. The light beam of rays within the apparatus is able to redirect it through the use of mirrors, and it is also able to be scattered with the assistance of collimation. Now let us discuss the difference between single beam and double beam spectrophotometers. One type of spectrophotometer known as the single beam spectrophotometer is an analytical device in which all the light waves emanating from the light source pass through the sample. Because of this, the intensity of light both before and after the light passes through the sample is what is measured. These single beam spectrophotometers have a smaller footprint and a more straightforward optical design compared to their double beam counterparts. Additionally, the cost of these instruments is more reasonable since it utilizes a single light beam that is not split in any way. The sensitivity of the direction of light beam after it has passed through the sample is very great. As a result, high energy exists throughout. Single beam spectrophotometers are available for use in analysis at wavelength ranges ranging from visible to ultraviolet. A single beam spectrophotometer is a type of spectrophotometer that determines the concentration of an analyte in the sample by determining the amount of light that is absorbed by the analyte in question. This is the point at which the Beer-Lambert law takes into the effect. According to this equation, the Beer-Lambert's law's equation, the concentration of analyte is directly proportional to the absorbance of the substance. An analytical equipment known as a double beam spectrophotometer divides the light beam into, that is, originating from the light source, into two distinct portions. While one portion serves as a standard, the beam used for the reference, then the other portion is used to analyze the sample, that is, the beam used for the sample. As a direct consequence of this, the reference beam is unable to travel through the specimen. The beam specimen has the capability to quantify the degree of light absorption exhibited by the sample. The absorption by a sample can be measured by comparing it with a reference beam. Hence, the absorption can be defined as the quotient of the sample beam which has traversed the sample and the reference beam. The spectrophotometer employs a monochromator to selectively isolate the desired wavelength from a light beam. Upon recombination, the reference beam and the sample beam proceed towards the monochromator. As a result, this approach mitigates or counteracts the electronic and mechanical perturbations on both the sample and reference beams in an equitable manner. The basic difference between the single beam and double beam spectrophotometers lies in the respective optical configurations. Specifically, in a single beam spectrophotometry, the sample is exposed to the entirely of the incident light beam, whereas in double beam spectrophotometry, the incident light beam is bifurcated into two separate beams with only one of these beams passing through the sample. Spectrophotometers are scientific devices utilized for the purpose of measuring the concentration of analytes present in a given sample through the application of a light beam. Consequently, this methodology quantifies the light absorption of the specimen. Advantages of double beam spectrophotometer. More consistent detection. Recent advancements in optics have facilitated a significant degree of automation and provide comparable, if not superior, levels of detection in contrast to previous single beam systems. The real-time measurement is not affected by instability factors such as lamp drift, state light, and voltage fluctuations. Minimal or negligible warm-up time for the lamp is necessary. This practice not only enhances the rate of output but also preserves the longevity of the lamp. The utilization of a double beam spectrophotometer offers significant advantages, notably the quick and consistent acquisition of measurement. It is not imperative to modify the transmittance within the range of 0 to 100% for every wavelength. Simultaneously, it exhibits the ratio of intensity between the beams of sample and the reference. Efforts are made to minimize the fluctuations in the radiation source. This technology enhances the efficiency of scanning over a wide range of wavelengths. Some limitations of double beam spectrophotometer. It is quite expensive. The use of a double beam spectrophotometer presents a considerable challenge. Application of double beam spectrophotometer. The utilization of a double beam spectrophotometer as opposed to a single beam spectrophotometer can prove advantages for various applications that necessitate stability, flexibility, and speed. The aforementioned tools are utilized within both research and clinical laboratory settings for the purpose of wavelength scanning, protein analysis, DNA analysis, quantitative analysis, and kinetic study. Double beam spectrophotometers are utilized by physicists, biologists, and chemists to quantify visible near-infrared and near-ultraviolet radiation. Summary of double beam spectrophotometer. The double beam UV visible spectrophotometer is a scientific tool utilized for the purpose of quantifying the light absorption of a given sample. The working principle entails partitioning the light output emanating from the monochromator into two distinct beams namely the reference beam and the sample reading beam. The reference beam is utilized for the purpose of measuring the energy of the lamp, whereas the sample beam is employed to reflect the absorption of the sample. The recombination of two beams occurs prior to their arrival at a monochromator, which enables researchers to determine the existence of analytes within a biological specimen by evaluating their absorption characteristics. The apparatus employs the principles of light dispersion, reflection, and refraction precisely gauge the absorption properties. The utilization of double beam spectrophotometer is feasible for the analysis of organic compounds through the assessment of their radiation absorption capacity. In short, the double beam spectrophotometer facilitates the transmission of light rays that result from the reflection process. The utilization of a single beam spectrophotometer is analogous to that of a double beam spectrophotometer. The biological specimens are illuminated by a light source and subsequently the results and are digitized through a computer system.